And because cell division is essentially just dividing in half, it is actually exponential growth. So when you study exponential growth in math, you know that you can get large numbers of cells dividing large amount of cells through very few cell divisions, right? So it's usually, if you look at the value, it's 2 to the n, where n is the number of cell divisions. Right? So if you have 2 cells undergoing 2 divisions, right, that would give you 4. But if you do this to like 2 to the 50th, right, that's like over 1 trillion cells. So cells, 1 cell, we can have, as we all start out at 1 cell, if those cells then divide and you follow them along, how many divisions? You only really need 50 cell divisions in all of them to produce the number of cells we have in our body. And so this seems to be kind of a common total number is 50. Total number of times a cell can undergo cell division. And so we could ask, well, what is keeping track of how many times our cells divide? And this has to do with the mitotic clock. So we have a mechanism by which our cells, our DNA, can tell how many times it has gone through cell division. And this has to do with the structure of the chromosomes. So this is a chromosome. Remember that I mentioned that chromosomes are DNA wrapped around protein. It is really tightly wrapped in the center, and this is called my centromere. So that's just a tightly wrapping of the DNA around the proteins. And at the very end, we have what are referred to as telomeres. So the very end, this would be a telomere. And on the other side, there would be a telomere too. And what they've discovered is that telomeres are sequences of DNA that are highly repetitive, that do not code for protein, so that as the cell undergoes cell division, it starts to lose its telomere. So every time it undergoes cell division, it loses pieces of its telomere. Okay? So when a cell divides, it loses some of its telomere. When no telomere is left, it's counted down. So when none is left, the cell can no longer divide. So the clock has been counted down. The cell cannot divide. So something interesting has appeared when we look at people of different ages and their telomere length. It seems to be very variable. So if you took like 10, 50-year-old individuals, they would have different amounts of telomere. So their cellular age, their chromosome age, differs between them. And so what they've actually discovered is that certain things cause your mitotic clock to tick down faster. And one of these is stress, right? So if you're in a very stressful situation, specifically even as a child, if you come from an abusive family, you are going to have a smaller amount of the telomere left when you're 50 than if you weren't in an abusive family. If you do drugs and don't exercise and have a poor diet, your telomeres are going to count down sooner. And so this is what they call chromosomal age. Right? So you could take and do a test. There are companies that test for telomere length. And so one of these is what is called telo years. So this is, they would like a sample of your DNA so they can look to see your telomere length. And then what they want to do is they want to see, they want you to do probably a bunch of surveys so that, you know, how do you eat? How much do you exercise? What do you weigh? What was your past, right? Have you ever smoked cigarettes? So they're looking at that because this might be kind of the key to aging. Right? So one of the interesting things is that we thought that telomeres only counted down. But what we've discovered is that there is an enzyme called telomerase. Oops, I can't write on that one. So telomerase is an enzyme that adds telomeres back on. So telomerase, remember I said anytime you see that ACE ending, right, that means that it's enzyme, like lactase is an enzyme. So this adds telomeres back on. So this would actually be something that you could do your extra credit on. Remember at the very beginning of the quarter, I gave you a handout in lab that said you could research a biotechnology company. So this would be something you do in telomeres, actually has a lot of genetic videos and genetic information. So they're actually trying to educate as well as do scientific research. So the telomerase is interesting because what they've discovered is that if you, say for example, if I decided that I was going to change my lifestyle dramatically, so I could send in my sample, they could take the sample, they could find out where my telomeres are, and then I could like stop drinking, stop smoking, and start exercising. And then they would measure your telomere length again to see if there has been a change. And so that's kind of the kind of data that they would like to collect. Okay. So you see in the news, right, that lifestyle changes might actually lengthen your telomeres. Right? So diet, meditation, exercise can improve the telomere length and reverse aging. However, if you live a very stressful life, you could actually have your biological clock. So you might know people that are like, they lose their spouse, and within 10 years, it looks like they've aged 20. And that could be due to the excessive stress causing a dramatic increase in the shortening of the telomeres. So when telomeres shorten, then your cells cannot reproduce, and so the tissues start to decline in their ability to repair and regenerate, and then eventually you succumb to old age and death. Essentially. Are there any questions about telomeres or telomere length? We're going to talk about cancer in a minute, and one of the things about cancer cells is that they turn that back on, and that telomerase back on, that gene for that, and they do not age. So cancer cells can become immortal. Okay. So we're going to talk briefly an overview of DNA synthesis. We're going to talk more about this when we get to molecular genetics. So when we look at our chromosomes, remember that we have 23 pairs of chromosomes. We visualize them something like this. We can actually take a picture of them using an electron microscope. This is an unreplicated chromosome. Which is why it might look a little bit different to you than what you sometimes see, which looks like this. Okay. So when the genetic material gets copied, the chromosomes turn into this, and so now there's two sets of genetic information. So if I put a gene on here, say like this is A, right? This is my gene A. It would faithfully copy this. So I would have two copies of the gene. So this is what is said to be a replicated chromosome. You need to be able to identify them as replicated versus non-replicated, unreplicated. So when we look at the replicated chromosome, it actually consists of 2 sister chromatids. So these are called sister, again, not because of the sex, right? These are sister chromatids. And so these are what are going to be pulled apart during cell division, so that each of the cells that are produced from the one parent cell are going to have a complete copy of the chromosome and the DNA. So that is what happens at the chromosomal level during DNA synthesis. Okay, so remember that DNA synthesis occurs prior to mitosis, prior to the G2 phase, where more cell growth and everything that needs to be produced for cell division is going to occur. So next we're going to talk about mitosis. So mitosis, remember, is asexual reproduction. It's cloning, and it has phases. And these phases are not really distinct from one another, but it would be like a set of dance moves, right? They just kind of go from one and then they morph into another phase. And so I'm just going to give you the phases that you need to know. There's more information in your book than you need to know. Okay. So the first that we're going to talk about is interphase. This is when the cell is not in mitosis. Okay. And so there are no chromosomes. You can't see the chromosomes because the chromosomes have not yet, the chromatin has not condensed into chromosomes yet. Okay. So the first phase of mitosis is, I'll put mitosis here again, just to clarify, is prophase. So pro means first or before. So this is where the chromatin condenses into chromosomes. The chromatin is just loosely wound, string-like substance, and it condenses into chromosomes. Now, the chromosomes are already replicated, right? So they would be condensed into replicated chromosomes. Okay. Here, also, the nucleus is disassembled. So it's taken apart. The nucleus is a membrane that is taken apart because we need to divide the whole cell, not just the nucleus, right? So all the organelles and cytoplasm and everything else in it is going to divide. Okay, the next phase is called metaphase. And meta means middle. That's how I remember this. And this is where the replicated chromosomes line up in the middle of the cells. Now, what is causing them to line up is the cytoskeleton. So remember, we talked about the cytoskeleton made of proteins that helps to transport substances. So the cytoskeleton helps to line them up, and it attaches via spindle fibers to the centromere. So the cytoskeleton attaches to the centromere. So that's the middle of my chromosome via spindle fibers. And this is actually what is going to pull the chromosome apart. So that is what happens in anaphase. Pulling apart of the chromosomes. So the sister chromatids get pulled apart. By the cytoskeleton elements. They get pulled to opposite ends of the cell. And then the next stage, the very last phase, is called telophase. And telophase is where the nuclei are reassembled around each set of chromosomes. And then we also have the dividing of the cytoplasm. So the cytoplasm divides. And this is called cytokinesis. So cyte means cell, kinesis means separating, right? So cytokinesis happens when the cytoplasm divides into half. So if we were to look at a diagram from your book, they have simplified our DNA. Remember that we have 23 pairs of chromosomes. Notice here that they just have 3. They have 3 chromosomes, 3 sets of DNA, right? And so prophase, the chromosomes condense. The nucleus gets taken apart. Ignore prometase. Metaphase is when they line in. These are the spindle fibers right here, spindle fibers. Anaphase, they get pulled apart. This is the new nucleus that is being formed in telophase. And then cytokinesis happens when you get a separation of the 2 cells. So I would put cytokinesis in telophase. Some people say that it's outside, but I'm going to put it at the end of telophase. Okay, so it's actually much more detailed than this. So let me see if I can get because we have more chromosomes, but we can actually observe this. So this looks like, I started over. This is underneath a microscope, so we can see what is happening. So the chromatin is starting to condense into chromosomes, starting to line up in the center. So these are replicated chromosomes. So you can kind of barely make out the 2 sides, right? And then this would be anaphase, where they're being pulled apart. And then you start to see cytokinesis happening, the cytoplasm getting separated. This is just showing it fluorescing. So this shows anaphase lining up. So we can observe how the chromosomes behave, just video after video. Okay, so the opposite of mitosis is apoptosis, or apoptosis, how I say it, apoptosis. And this is programmed cell death. So we now know that mitosis is really important in development and regeneration of tissues, but also programmed cell death is really important as well, specifically like during embryonic development. So for example, when our fingers first form, we have webbing between them. Those cells must die in order for us to get fingers. Sometimes people are born with the webbing still in place, right? And so it makes it hard for them to use their hands or their toes And so they will actually cut them and remove the extra tissue, right? So this programmed cell death is different than cells actually being eaten by white blood cells when they're damaged. These cells undergo cell death in order to better support the multicellular organism, okay? So this would be like during embryonic development. Certain structures disappear. It's also important in the development of our immune system. So we kind of randomly produce these blood cells called T cells. And these T cells, if they detect our body as being foreign, they will undergo apoptosis. So the development of immunity, so the development of the immune system is another place where apoptosis is very important. So cells that cannot recognize normal body cells will die. This actually takes place in your thymus gland. And it's actually a type of they believe it's like cell suicide. The cells will just release their enzymes to the inside, and they'll just start to digest themselves from the inside out. So it's really nothing coming in, attacking them. They just undergo programmed cell death. There's probably other places where this is really important, but this is kind of an area of research that is new, the study of apoptosis. Okay, so let's talk about cancer. So cancer is unregulated mitosis. So this means that the cells divide inappropriately when they should not, and they also sometimes create masses of cells that are referred to as tumors. So we could talk about the characteristics of cancer cells. Okay. The first one is that they have abnormal DNA. Abnormal DNA, including chromosomes. So they might have extra chromosomes, for example. Their chromosomes might be broken, right? So essentially, they have mutations. Right? And so chemicals that cause mutations that lead to cancer are called carcinogens. So tobacco smoke is an example of a carcinogen that could cause mutations in the DNA, which could cause the cell to become cancer. So mutagens that cause mutations that cause mutations are carcinogens when they cause cancer. Right? And sometimes you see this on like paint cans or whatever. In California, they say, in California, this is a known carcinogen because California has the strongest environmental regulatory of all the states, right? So they have strong regulation that anything that's a carcinogen has to be labeled as such so that you are careful when you use it. The second thing about cancer cells is that they do not stop dividing when they become too crowded. Normal cells, if they become too crowded, they stop dividing. So we say that there is no contact inhibition. Right? So they will divide even when crowded. So we also call this density dependent, right? They do not stop when their density becomes too high. So this is density dependent. Right? So this is why they form masses and they form tumors. Right? So those are masses of cells that are very crowded together. And so tumors could actually be benign. So benign means that when they look at them, their DNA appears to be normal and they are not cancerous. So tumors, benign tumors, are not cancerous. So sometimes those will form, right? But then if they're cancerous, what are they called? Malignant. Malignant means they are cancerous. They sometimes actually, when they become cancerous, they can actually they release chemicals that cause blood vessels to be formed to feed them. So they actually cause special blood vessels that go right to the tumor that feeds the cancerous cells. And that's another sign you can see that they're malignant is they cause those blood vessels to form. So they're using up nutrients that are needed by other cells. And so sometimes in cancer, people get really thin because the cancerous cells are using up all the nutrition that they're taking in for their own growth and reproduction. Okay. They also have no apoptosis. So they do not undergo programmed cell death. They have no mitotic clock. So theoretically, they are immortal. So theoretically, they could go on dividing over and over and over and over again. So there is a group of cells that we have been using as study cells for over 50 years, and these are called HeLa cells. Who in here has heard about the HeLa cells? This is another something else that you could do your research or your extra credit on. HeLa stands for a woman named Henrietta Lacks. And Henrietta Lacks got cervical cancer, and so she went to the Johns Hopkins hospital where she was treated for free. She was really interesting, the way that they treated cervical cancer way back in the 1950s was that they actually used radium. So we talk about radium because that's the isotope that emits radiation, right? And they painted it on watches so that they could see the watches. So radium was actually inserted into her vagina to try to kill the cancer cells, the cervical cancer cells. But Henrietta Lacks died when she was 51 of cervical cancer. Actually, she died in 1951, sorry, died in 1951. I think she was in her 30s from cervical cancer. So back then it was, I guess, okay to do experiments. And so they took the cancer cells after she died, they took them out of the body without anybody's permission from the family. And then what they started to do is they started to grow these HeLa cells in petri dishes. And they discovered that the HeLa cells are really, really, really good at reproducing. And it was the first cell lineage that they could actually grow in bottles, and the cells would not die. And so pretty soon, they were sending these cells all over the country and then all over the world. And people were doing research on HeLa cells for just studying cells in general, but in specific, understanding how cancer cells differ from normal cells. So we're just going to watch a short little video that talks about this HeLa, Henrietta Lacks. It is rather controversial these days because her family did not know anything about the use of these cells. And so if you search HeLa, you find all these videos. There was even recently a documentary made about it. And there is also a book called The Immortal Life of Henrietta Lacks. And so this is a picture of her before she became sick. And this is a short video talking about the significance of those cells.
Something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor, dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week, and scientists put them to work in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers. and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.
Okay, are there any questions about that idea? So those HeLa cells were so prolific that they invaded other lineages, and then people had to reconsider their research because they didn't realize that the HeLa cell had taken over like their normal pancreatic cells that they thought they were studying. So they actually had to go back and revise their scientific findings to say that we were actually studying HeLa cells. So they're actually quite invasive in the lab because their ability to stay on glass and then to also float through the air and then to get into other tissue cultures. Okay, so when we look at another characteristic of cancer cells that is important is that they become unspecialized. So cancer cells, 5, which would be the 5th characteristic, cancer cells go from being specialized to unspecialized. So they become unspecialized. So what that means is that they can live anywhere in the body. So like you could talk about a skin cell becoming melanoma, and that melanoma, that skin cell, could then travel through the blood to the brain, and then it could start to develop a tumor in the brain because it is unspecialized. So this means they become unspecialized and can travel via the circulatory system to invade other parts of the body. This has a name where the cancer has, what has it done if it gets into the circulatory system? What is the word? Does anybody know? Metastasized. Excellent. Oops, can I spell that? Metastasized. Okay, so metastasis is a really bad. So if your cancer is in one spot, they might be able to go cut it out. They might be able to radiate it. But if it gets into the circulatory system, then you have to use chemicals that also can travel via the circulatory system to try to kind of shut down the cells that are reproducing. And that includes all of the cells that are reproducing, including the ones that produce your hair. So chemotherapy oftentimes kills those cells, causing your hair to fall out, because it's actually killing all the cells that are currently dividing in the body. Okay. So I'm just going to briefly introduce meiosis, because this is something that we're going to be looking at in the lab. Okay. So meiosis is sexual reproduction. And it is the production of what we call haploid cells. So haploid cells have half of the genetic material. And so this would be the egg and the sperm. This only takes place in the ovaries and testes. And it is a special type of cell division that has 2 consecutive cell divisions. So we have 2 cell divisions that happen one right after the other. And these are labeled mitosis 1 with a Roman numeral, and mitosis 2. Okay. So if I was to draw a cell, this would be like a cell in the testes that is getting ready to divide. It would divide once, and then without replicating the genetic material, it would divide again. So what you have is this initial cell, this parent cell, has the full component of the genetic material and the chromosomes are replicated. In this, they don't have the full component of chromosomes, but here they have just half the number of chromosomes. So these are the haploid cells right here. Now, the biggest difference between mitosis and meiosis comes from how the chromosomes line up in the first metaphase. So we can talk about metaphase 1 of meiosis 1. So in this first phase of first metaphase, the chromosomes line up in pairs. So I'm going to draw my cell, and I'm going to have my chromosomes. They're replicated, but they're lined up in pairs. Right? So instead of the chromosomes being pulled apart at the sister chromatids, what's going to happen is that these chromosomes are going to be pulled apart, but they're not yet going to be replicated. So I can put my spindle fiber here, pulling it to opposite ends. And so you can see that after these get pulled apart, the genetic material is still duplicated. So then it is in my metaphase 2, the second cell division, that the sister chromatids actually get pulled apart. So that's metaphase 1. So if I just show you an overview from your book, we'll talk more about this in a little bit of detail on Wednesday. Okay, so these are my chromosomes. They're blue and red because we're referring to the ones that you inherit from your mother and the ones that you inherit from your father. But notice how these replicated chromosomes line up and then they're pulled apart. And then during the second cell division, the sister chromatids get pulled apart. And notice that at the end, I no longer have chromosomes that are replicated. And I also no longer have pairs of chromosomes. So that if two of these came together, say this was an egg and this was a sperm, then each parent would contribute one of the chromosomes to the other. And so you would have pairs of chromosomes coming together after fertilization of the egg. So we'll talk more about this. And this is really actually kind of important to understand when we get to genetics. So this is kind of a lead up to doing genetics problems and understanding how traits that are caused by genes on chromosomes are passed from one generation to the next. Okay, so we'll finish there. And for those of you who have Monday lab, I'll see you in a half an hour